Hello everyone, welcome to the online lecture. I am Parameshwari, working as DL in Botany, TTWRDC Devarakonda. Today I am going to deliver an online lecture on the topic secondary growth in a dicot stem. This is how a stem after the secondary growth happens, how it looks. Stems. First, we'll start with stems. So, this is the simple structure of a plant body. Now, stems are the organs of a plant which grow above the ground. Now, these stems support leaves and fruits. The function of the stem is to conduct water and sugars throughout the plant with the help of conducting tissues, xylem and phloem. Now, here you could see two different types of stems. You can observe here the one which is on the left side has very thin stem compared to the one which is on the right side. Do you observe any difference? Exactly. The right side picture is having a bigger diameter comparative to the left one. So it is the thinner one is the herbaceous stem, and the one that is the tree, it is a woody one. Now, before getting into the topic, let's see the role of cells. What are they? What level of organization do they? And we are going to study about the tissues which play a major role and support the plant body. Now, the plant cells are the basic building blocks. They specialize in form and function. By working together, they form tissues and they can support each other and survive. The levels of organization, atoms form molecules, molecules into cells, cells into tissues, tissues into organs. The whole organs become the whole plant. Now, let's take a look of the plant organs. Organs are nothing but the tissues that act together to serve a specific function. Here you can see all the three organs of a plant, starting from the base, roots, stems, and leaves. Here you can see the types of stem showing the internal structure of a monocot stem, dicot stem, and a root. Now here you can see the difference in the difference as well as how they are arranged in the internal structure of the different stems, monocot and dicot stem, and the root. The stem anatomy, here you can see the outermost layer, epidermis, cortex, and pith, and in the middle, the vascular bundles are represented in a violet color, ring-like structures. Let's take a look at xylem, which is a water-conducting tissue, and it consists of different elements like tracheids, vessels. These are tube-like structures. They play a vital role in the plant body. Next, we'll see the other type of conducting tissue, that is phloem. Phloem transports sugar. Phloem also consists of different elements Phloem is composed of sieve tube members, companion cells. And here you can take a look at the diagram of the phloem, which will give you an idea to understand the secondary growth, how the phloem is arranged in the secondary tissues. Now, here if you see, this is a like cross section of a stem where you can see xylem and a phloem, which we have discussed just now. And the layer, the strip present between the xylem and phloem is a cambium. Together constitute vascular bundles. Now we see different types of vascular bundles in the plants, simple and compound. Now in compound, we'll see concentric and collateral type. Collateral open when the xylem and phloem are separated by cambium. If cambium is absent, then it is called a closed type of vascular bundles. Now, as our topic is about dicot vascular bundle, so comparatively observe the dicot and monocot vascular bundle. Now, if you see the enlarged vascular bundle, one of the vascular bundle among the ring, you can see the primary xylem, metaxylem, and the vascular cambium, which is present in between the xylem and phloem. Now take a look at the anatomy of Angiosperms vascular bundle system. See how the water is transported through the xylem and the phloem, and as well as the internal structure of dicot and monocot stem. Now, coming to our topic, what is secondary growth? Secondary growth is an increase in girth or width of a plant initiated by cell divisions in lateral meristems. To be short, secondary growth is nothing but the increase in the thickness. Now, all plants, not all plants, but some of the plants show secondary growth due to the activity of vascular cambium. Now, here you can observe how a primary growth, a plant body with a primary structure and a plant body with secondary growth, you can see the different layers and the additional layers formed after the secondary growth. Now, before discussing about the secondary growth in depth, we have to know about certain tissues. Okay, the group of cells are nothing but the tissues which play their important function and role. Now, these plant tissues are of meristematic tissue and permanent tissue type. Meristematic tissues are again of apical meristem, lateral meristem, and intercalary meristem. Permanent tissue consists of simple and complex. Now, the xylem and phloem, which are represented here, are the main focus of the topic. Since the secondary growth means the formation of secondary xylem and secondary phloem is the part of this secondary growth. So we need to know the what are xylem and the phloem. Now, see the primary structure of a plant. Observe, you can see here at the shoot tip, you can observe certain meristems called apical meristems, which we have seen in the last slide, which are represented in the last slide. Now, this apical meristem is responsible for the growth in length of this shoot, and as well as the lateral meristems, which are present towards the sides of the plant, the vascular cambium and the cork cambium, which play a major role in the secondary growth and stems. Okay, next, the root apical meristems are also shown here. Now, a clear differentiation between primary growth in stems and secondary growth in stems is to be visible here. You can see the increase in number of layers and the additional layers comparatively with the primary and the secondary growth in stems. Now, if you see, and the primary growth of the stems, epidermis Sorry for the inconvenience. There was a small disturbance in the internet connection. And continuing, now we are in the secondary growth and stem, seeing this different layers formed after the secondary growth. See, the secondary xylem and secondary phloem are formed, and also cord cambium is to be visible. And in place of epidermis, periderm is also to be seen. So we'll take the example of Helianthus annus, which is a sunflower. The tears of this Helianthus annus sunflower plant stem, we'll look at it and we'll observe the primary structure of this Helianthus annus. Okay. Now the primary structure of a dicot stem, a typical example is the sunflower stem. It consists of different layers. It is usually differentiated into three distinct zones: epidermis, cortex, and endodermis. As you can see here in the ground plan, it is represented here. The second label, and the cortex, which is represented in light blue color, and the endodermis. Now, starting from the first zone, epidermis. This epidermis is a single layer, outermost layer present on the surface of the surface of the cells, and it is covered by cuticle. If we observe here, the epidermis is that epidermal cells are arranged compactly without any intercellular spaces. Now, in these cells, there could be some chloroplast present, and epidermis gives rise to epidermal hairs. Next to epidermis is the hypodermis. Hypodermis is immediately present just below the epidermis. Okay, this hypodermis is part of the general cortex. This hypodermis is having 2 to 3 layers, and the chloroplast chloroplasts might not be present or absent. They are made up of colon cells. After hypodermis, we can see the next structure is the uh, general cortex. It is of several layers and consists it made up of parenchymatous cells. After that, we see pericycle. Pericycle is the last layer of this general cortex. Okay, now moving on to the third zone, that is the stele. Stele consists of the endodermis and vascular bundles and pith region. Now, this endodermis is made up of barrel-shaped cells. And next, the vascular bundles are nothing but the combination of xylem and phloem, which you can see here. And these vascular bundles may be 15 to 20 in number, and they are arranged in the form of a ring. Usually, these vascular bundles are endarc, open, collateral, and conjoint. The pith is present to the center. It is the innermost zone. Now, we'll move on to the secondary growth. Now, this secondary growth is because of the activity of the vascular cambium and core cambium, which are the secondary tissues, okay, which are formed in the later stage of the growth of the plant. Now, this is how a vascular cambium looks. You can see here the labeling which is present between xylem and phloem. It occurs generally in woody stems, and this the position of it is between xylem and phloem and the middle of the xylem and phloem. Now, as we said that the secondary growth in some plants is because of the activity of vascular cambium. This is a type of secondary tissue. Okay, and in plants usually usually the primary activity is primary structure of the plant is because of the apical meristem, which we have just now seen in the representation, the chart about the meristems. Now, and in some monocots, in some dicotyledonous herbaceous plants, secondary growth is not to be seen, but in some woody gymnosperms and angiosperms, secondary growth happens after primary structure is formed, and the activity of this primary structure is only for a limited time. Subsequently, these secondary tissues are formed when uh, the plant resumes to the activity of secondary growth. In secondary growth, the width of the plant increases. Not only the height of the axis increases, the width of the plant also increases. The increase in the width of the plant is because of the activity of this secondary meristems, which are the vascular cambium and the cork cambium. Now, this vascular cambium gives rise to secondary xylem and secondary phloem. And the cork cambium gives rise to periderm. This cork cambium is formed by the differentiation of the hypodermal cells, which is present in the cortex, and it gives rise to the periderm. Periderm constitutes cork, uh, cork, secondary cortex, and the phloem. Here, if we observe the two vascular bundles, means the xylem and the phloem. Okay, the phloem, which is present towards the upper side, and the xylem towards the lower side. In between these xylem and phloem, some meristematic cells are present. Those are called vascular cambium. Those are formed from the procambium. And in between this vascular cambium, medullary rays, the cells of the medullary rays are present, which actively divide and form a strip between the two vascular cambium. As a result, they join together and form a cambial ring. As you can see here, the intervesicular cambium, which is labeled here in the diagram between the two vascular bundles, are joined together. Now, if we see here, here we can observe the epidermis, cortex, next the primary phloem, which is present towards the outer side in the diagram A, and primary phloem towards the inner side, and in between those two fascicular cambium is present. Now, after the development of this activity of the cambial ring, uh, after the development activity of this medullary ray cells, as a result of which the intervesicular cambium was formed, and the intra and intervesicular cambium join and form into a cambial ring, as you can observe it in the diagram B. And in the secondary growth, you can see here the primary xylem, how it is being crushed into the center, as well as the primary xylem is crushed to the center, and the primary phloem is ultimately stripped off because of the development of the later form secondary tissues called secondary xylem and secondary phloem. And also, epidermis is being replaced with another layer called periderm. and the vascular cambium is to be clearly visible. Let's look into the growth pattern in wood and bark. Now, the continuous source of food in a tree is due to the presence of this vascular cambium. Why? Because of the activity of this vascular cambium, secondary xylem is produced. This secondary xylem is nothing but the wood. So the vascular cambium produces the secondary phloem that conducts food within the inner part. Two types of meristematic cells from the vascular cambium are formed. Those are the fusiform initials and ray initials. Fusiform, now these initials undergo periclinal and anticlinal divisions, because of which the secondary tissues are developed or formed. Take a look at the periclinal and the anticlinal divisions. Here you can clearly see here the periclinal divisions which are occurring in the parallel in a parallel way to the surface, and the anticlinal divisions are occurring in the perpendicular way. So as a result, you can see here the alignment and how the cells of the vascular cambium are given out, and as a result, new layers of cells are added to the vascular cambium to the inside and the outside. So we can observe the increase in the thickness of the diameter with the multiplication of these cells or differentiation of these cells. If we see here, now the cambium which is present between the xylem and phloem. Okay, the cambial rings start dividing, giving rise to cells both to the outer side and the inner side. The cells which are given out to the outer side are the cells of secondary phloem, and the cells which are given out to the inner side are the cells of the secondary xylem. Now, if we see here, the amount of secondary xylem is produced in large quantity compared to the secondary phloem. Now, here, initially, when the plant is in its primary structure, we can see how the vascular bundles are arranged and the procambium is present, and later on the intervascular and vascular cambium join and form the cambial ring, and we can see primary phloem and primary xylem. Next, after the activity of this vascular cambium, when the secondary phloem and secondary xylem are given out, they are they form ring-like structures, and the increase in the thickness of the stem also happens. So these primary xylem and the secondary xylem which are formed together form an first annual ring. Here you can observe the ray initials and the fusiform initials, which we are going to discuss in the later slides. Now, the fusiform initials are spindle-shaped and they are elongated cells. Now, based on the arrangement of the cells, they can be like striated or non-striated. Now, the cell divisions which occur in this fusiform initials typically produce more amount of xylem than phloem. Now these ray initials no more help in the conduction but give mechanical support to the plant. Fusiform initials give the mechanical support. Now, the other type of initials are the ray initials. These are the horizontal initials which are isodiametric in shape. Okay, the main function of this ray initials is to conduct food material. Now, here you can clearly see the three-dimensional view of vascular tissue in a stem. See the primary xylem present towards the inner side and the primary phloem towards the outside, how the vascular cambium and the ray and the fusiform initials are arranged. Now these vascular tissues form rings in trees. Okay. Now in a season, in 1 year, the vascular tissues which are produced in the two growing seasons together form one annual ring, which is counted as the age of the plant. So, annual rings are the xylem formed by the vascular cambium during one growing season. Therefore, the one ring is equal to 1 year. This is how a secondary growth activity produces wood. That is the secondary xylem towards the center, and how the xylem and phloem are crushed and the bark is replaced with the epidermis and the vascular cambium is clearly visible. This is a microscopic picture of the annual ring of a plant stem, epidermis, phloem, and an annual ring. Two rings which form its age. Now, let's look at the wood. The wood is produced in the secondary growth activity of the plant stem. Now, the wood is the secondary xylem of the seed plants. Now, a tree trunk is composed of the following basic parts listed in order beginning with the outer layer. Now, the wood is of two types in the plants. It is the heartwood and the sapwood. The one which is present towards the center and is called heartwood, and the one which is light in color and which is present towards the periphery of the heartwood is called sapwood. Here you can clearly see how a woody stem looks like, starting with periderm and secondary phloem, vascular cambium, sapwood, and heartwood. Let's look into the heartwood. Heartwood is also referred to as duramen. This heartwood is dead material, and it is dark in color. This consists of many substances like tannins, resins, gums, and they have commercial value, and it gives mechanical support to the plant. Now, the main function of the hardwood is to provide support to the tree itself. Now, sapwood, it is also referred to as alburnum. It is a light-colored wood present outside of the hardwood, and it its function is to transport the food materials. Next is the cork cambium, which is another type of secondary meristematic tissue. Now, this is formed with the pressure created by the intrastellar secondary growth. That is, the secondary xylem and secondary phloem are formed, are keeping on adding to the to the stem. Therefore, it creates a pressure on the epidermis and the and the lower hypodermal cells. Therefore, this cork cambium is formed in the hypodermal region. Here you can see the structure of cork, hardwood, sapwood, then vascular cambium. After the vascular cambium, the layers whichever are present comes under the bark, and how a cork is formed from the activity of the cork. Here, the right side diagram, you can clearly see the phelogen and the phellum and the phellodum. In this, the phelogen is a cork cambium which is formed first, and it cuts off the cells towards the upper side and the inner side. We'll see the development of peridome. Peridome is a word for the three layers or cambium, cork, and secondary cortex. Now, this peridome is formed by the differentiation of one of the layer of the hypodermal cell, which resumes its meristematic activity and forms into a cork cambium, as it is represented in a brown color here, you can see. And this cork cambium, after resuming the meristematic activity, gives the cork cells towards the outer side and the phylloderm to the inner side. And later on, the number of cork cells are going on adding, and they increase in number, and it is the cork, and these cork cells are suberized. The cells are surface is coated with suberized, and they become thick and impervious to gaseous and gaseous substances. Here is a microscopic picture showing epidermis, how the epidermis is stripped off with the formation of this peridome in the cortex region, cork cambium and the cork formed. Now, here, next, is the combination of layers consisting of all the layers above the vascular cambium. Here you can see the soft type and hard type of bark. Soft is the first formed one, hard is the later formed one. Now, bark is having two different regions, inner bark and outer bark. This bark appears to be stripped off, and because of the splitting of the older layers of the periderm. Now, these lenticels are the lens-shaped structures which are formed in the periderm for the exchange of gases, as the suberin deposited cork cells are impervious to gases. Therefore, loosely arranged cells are formed, which are called complementary cells, which allow the exchange of gases and respiration process and photosynthesis to take place. This is how a lenticel looks on a tree. In the stem. So finally, if we see here from the above, how the secondary growth is been taking place in the stem. If we observe here in the very first, the topmost one, we see primary xylem, primary phloem with vascular cambium. And later on, after the activity has slowly started, secondary xylem and secondary phloem are given off. So a little bit of growth has happened. Now, after the complete, after many years of the secondary growth, we can see that here the bark or epidermis has been removed and the periderm is formed, and a bark is formed, periderm with 3 layers, secondary phloem, cork cambium and cork, as well as cork cortex, secondary cortex. Now, the wood and bark has many applications, commercial values. Now, the wood has two main uses, mainly as a fuel and paper production. The wood pulp is used as paper. It is nothing but the thin film of the cellulose made from it. You can observe the wood pulp here in the diagrams. Notice the paper is usually made from the white spruce here. It is extracted from the plant Picea glauca. Now, the wood consists of, as we have seen that the wood, in the hardwood, we have seen many substances which are deposited form like latex, resin, and the oils and all. They also protect the plant and as well protect the plant from diseases and from insects. So here you can see how the resin, the gum, mucilage, and the latex extracted from the plants. Here, a latex from the chicle and willow tree is used in chewing gum. So thank you. That's the end of this lecture. Thank you, everyone.